Hey everyone, so today in this video we'll be talking about the ratio which cells maintain the concentrations of ATP and ADP and how cyanide works to set this balance. So as we've learned in class over the last few months, hydrolysis of ATP is an exothermic process. This means that it releases energy after it's completed. This occurs because as water interacts with ATP, the bond between the gamma and beta phosphate group right here is broken, which releases a lot of energy. This results in a very large delta G value, or Gibbs free energy value, of around negative 30 kilojoules per mole. However, as the concentration of ATP rapidly decreases in the cell, and the concentration of ADP increases relative to ATP, this delta G value approaches 0. Also, when this happens, the reaction begins to become reversed, such that ATP becomes more concentrated again. In a typical normally functioning cell, the concentration of ATP will always be 10 times that concentration of ADP.
As long as the mitochondria are functioning properly, the 10-to-1 ratio of ATP to ADP in the cell will remain constant. However, substances like cyanide can work to disrupt this balance. This diagram shows the electron transport chain along the inner membrane of the mitochondria. Complex 4, also known as cytochrome C oxidase, is an enzyme that works to reduce oxygen and create water to contribute to the hydrogen gradient used by ATP synthase to make ATP. Cyanide forms a tight bind onto complex 4 and inhibits any contribution that it has to the hydrogen gradient, thus greatly reducing the ATP output. Since ATP will still be consumed by the cell, this greatly reduces the ratio of ATP to ADP and can lead to cell death.